All right. So the purpose of this lab is twofold. Okay? The first purpose of this lab is to get us some experience and practice using the microscope before we got to seriously look at the internal structures of cells. Okay? The second purpose of this lab is to look at how a microscope works. Okay? How does it in fact magnify and produce the images that we can use to see essentially small objects. Right? That's going to be the main purpose of the lab. That's the problem that we're investigating. Now, if you recall, yesterday I went over how light travels through a microscope and what happens to it. It might be valuable to revisit that, okay, as part of your analysis and stuff like that. Okay, for our design, okay, the experiment's going to work like this. We're going to look at a letter E cut out of a newspaper. Okay, we're going to look at an amoeba, a paramecium, and a euglena. Okay, we're going to look at 4 things. Okay, the letter E is obviously quite large, and it's a shape we're familiar with. Right? The purpose of looking at the letter E is essentially to tell us what happens to something when it's put under the microscope, because we know how an E is supposed to look. When we look through the microscope, it may or may not look different than that, and that can tell us a lot about what happens to the light as it comes from the specimen and into the microscope. So that's the purpose of looking at the letter E. Okay? Yes, a lowercase letter E. We're going to make a microscope slide called a wet mount with a letter E cut out of a newspaper. Okay? Pretty easy thing to do, but I'll go over the process of how we do that. The other things we're going to look at, an amoeba. An amoeba is kind of a gelatinous blob, okay, that lives in pond water. It's a single-celled organism, but it's fairly large for a single-celled organism. So it'll be easy to find, easy to get focused. Okay? We'll look at a paramecium, which is similarly, except it's not a blob. It actually has a fairly specific kind of hourglass shape, okay, that lives in pond water, single-celled, large enough that we'll be able to easily get it under the microscope and focused and identify structures. The last thing we'll look at is the euglena. It has a similar shape to the paramecium, but is way smaller, right? And that one is going to be kind of a test your skills, get this thing found, focused, etc., using the microscope. Okay? That's why we're doing so much of the pre-lab today, so that we have the whole period after the quiz tomorrow to find those specimens, get them focused, get good pictures of them, okay, things like that. All right, so our design. Our manipulated variable is, what are we changing? Every time I look through the microscope, I'm looking at something different. Okay? Why do I want to look at different things in the same lab? What is that going to tell me about the microscope? That's the thing you probably want to work into your explanation for why the specimens are the manipulated variable. Okay, responding variable. Well, that should be fairly obvious. Every time I look through the microscope at something different, okay, there should be a slight change in what I see. Okay, find a way to explain that. Controlled variables. Well, anything that's going to be the same every time I look at something different should be a controlled variable, right? So basically anything that is either a piece of equipment that is the same every time, or any method that you use that is the same each and every time we look at something. For the hypothesis, as always, if and then statements. Okay? The if part answers the question, the and part describes the experiment, the then part gives us predicted results. So the explanation you have here, generate a hypothesis about how a microscope works, path of light. Okay? That would be the if. The and part describes the experiment. Okay? I would say those would be things you would probably want to include in those 2 parts of your hypothesis. Okay, procedure, fairly straightforward. Get your microscope, get the slides. When we are over there tomorrow, you only need 1 slide at your lab bench at a time. Okay? Every time I allow people to take all of them over, somebody inevitably, while looking through the microscope, does this and sweeps them onto the floor where they break. Okay, so 1 slide at a time is all you need. You can come up and get the next one when you're done with the first. Okay? Make lab drawings of all of our specimens. Well, you're going to do that by taking pictures of them using your phone. Okay? Note on that, if you have a protective case on your phone, you usually have to take it off in order for the camera to get close enough to the lens for you to get a good picture. Okay? Absolutely. Yep. Yeah, one person can take the pictures. If they get good pictures and they can send everybody in the group, or each person can take their own, it makes no never mind to me. Okay, as long as everybody's got the pictures. Okay, and then each group member is going to create a wet mount of the letter E and view it under the microscope. All right, how do we make a wet mount? Wet mounts are pretty easy. You're going to get a microscope slide that doesn't have anything on it, so a rectangular piece of glass. Okay, and you're going to lay it down on the lab bench. You're going to cut out a letter E from a newspaper, a lowercase letter E from an article, not from a headline. We want it fairly small. Okay? And you're going to put that letter E that you've cut out, okay, with its jagged edges from the piece of paper, like this, okay, onto the slide. Then you're going to take an eyedropper and add a drop, maybe two, of water to the letter E. At which point the letter E will probably flip upside down, like facing the wrong way, and you'll have to play around with it with either a pair of tweezers or a probe or something like that until you get it soaked up, and then you can manipulate it the way you want. Okay? We want it to be sitting on the slide so it's right side up. Because that way, when we look through the microscope, we'll know if it doesn't look right side up what the microscope has done to the light. Everybody follow me? Okay? It is crucially important that the letter E is the right way. All right, so I've added the water. The letter E is now oriented the right way. Okay, so I've got like my letter E kind of sitting right here in, let's say, a bubble of water, right? Something like that. So now I'm looking at it from the side. I take a cover slip, which is a small square piece of transparent plastic. Okay, I set it on the slide right about here, and I drop it. And it falls down like this, and when it does that, it squishes the water to the edges and pins down the letter E. All right. Why do I bother putting a cover slip on? Well, it doesn't move around, and it also keeps that stuff from getting on the bottom of the slides, okay, or on the bottom of the lenses, I mean. Right? So it serves that purpose. Why do I bother using water? That's in the analysis. You'll have to figure that out. It might have something to do with that stuff we talked about water being special in the last unit. There may have been a question on your unit exam about the special things water can do that might hold a slide. Yeah, anyway, okay. So that's how you make a wet mount. Pretty easy. Stick it under the microscope, look at it, take a picture of it. When you're done, the cover slip and the letter E go in the garbage. You dry off the glass slide, put it back in the container. Okay, pretty easy for that. That's the first one you should look at. Okay, you should look at the letter E first. It's the biggest, it's the most obvious. Okay, gets you kind of familiar with the focusing procedure and all that kind of stuff. Okay, after that, okay, you'll be looking at other specimens. Oh, so there's the letter E. Okay, so these are the three specimens you're going to look at. This is the amoeba, the big pinkish purple blob. Okay, they all have a slightly different blob shape because no two blobs are alike, just like snowflakes. Okay, so it'll have some kind of blob-like shape. Okay, there'll be kind of a darkened area. This is like a nucleus. Okay, things like that. The only issue with the amoeba is that it is kind of big for a microscopic organism, and when you're focusing it, especially on high power, it's, even though very small, three-dimensional. And so what ends up happening is you never really get all of it in focus at the same time. Okay? And you can kind of see that here. This little area right here is part of the amoeba, but it's not as focused as this part because it's behind it. Right? There's this depth. Right? You probably haven't noticed this, or maybe you have, but if I'm looking at Shaz right now, she's in focus, but Kennedy is blurry, because that's not where I'm focused. She's further away. The same thing happens with a microscope if you have something that's large. Okay, the depth will kind of mess that up. So what you may actually have to do is take a few pictures of the amoeba, okay, and then just use whichever one is best to label it. Okay, but you may have to take a few and kind of focus through it to see all the parts. The other two, you won't have that problem with. They're much flatter. Okay, the paramecium. Okay, that's this one here. Okay, you can see these kind of blurry areas at the edge of both of the paramecium. Get up here, you can actually see they almost look like little hairs. That's what they look like. It's little hairs. They're not hairs. They're called cilia, And that's how the paramecium moves. Okay, it waves these things around and it can move really quickly. By the way, all the specimens we're looking at are long dead and preserved on the slide. So they do not move around. Be thankful for that. Okay, in university, we had to look at the paramecium. We had to actually like breed them, which is difficult because they're microscopic organisms. But you would have to do that. But you couldn't see, they were so fast in water that you couldn't ever see them for very long. So you'd put a drop of glycerin, which has the consistency of cold maple syrup, okay, onto the slide, and they would slow right down so that they couldn't swim through because it was thick, okay, and then you could view them.
So if they're alive, they're in water?
Yeah. But pond water, not fresh water. Yeah, and they're not going to hurt you. Okay, there are some amoebas that are parasitic. That's why you don't want to drink unclean water. Okay, you can get amoebic dysentery if you drink unclean water. That means you have diarrhea for weeks on end, and it kills a lot of people worldwide. Okay. All right. The last thing you're going to look at. The last thing you're going to look at is the euglena. Now, this is greatly enlarged. When you look at it, you're not going to see this much detail. It's a lot smaller than that. Okay? If you hold the slide up underneath the light, you might see tiny little blue slivers. Okay? In fact, sorry, you might see that on the paramecium slide. You will see tiny little blue slivers under low power, okay, of these things. They're that small. Right? Once you get them to high power, you'll see something that has roughly this shape. It doesn't have the cilia like the paramecium does. It has a long tail called a flagellum. Same way a sperm would swim, that's how they swim. Okay? They have a flagellum that they whip back and forth that propels them along. The cool thing about euglenas, they're mobile and they're photosynthetic. So they're a plant that can move. Yeah, which is pretty cool. All right? They're not really a plant, they're a protist, but they're photosynthetic and they can move around, which is pretty cool. Okay? So those are the three microscopic organisms that you will look at tomorrow. These will be included, these pictures, listening please, will be included with the assignment. They're not right now, but they will be included on there for your reference purposes. Okay? I do not want to see my pictures in your lab report unless you're absent, okay, on the day we do the lab. And even then, it would be nice if you got somebody else's pictures. Okay? If you're in absolute desperate need because you're not here, then you can use mine, right? But if you are here that day, you use yours. Okay? Questions on that? Eric. 3 controlled variables, yeah, same as always. Okay, remember guys, our lab drawing format, I keep this one because I'm very proud of my drawing. This is like my best drawing ever, okay? But remember, you're not going to draw it by hand. You're going to take a picture, okay? And make sure that all your labels go to one side, that you have your name up here. I know, it was a long time ago. I've kept it for a long time, okay? All right, and don't forget, title goes below magnification and size. What's that? It's exactly the same day. That's weird. Okay. That means I'm super consistent in the rate at which I teach this class. Okay. So we've covered, guys, we've covered basically everything we can do pre-lab. Okay. Tomorrow, after we've viewed everything, then you'll be able to answer the analysis questions. Okay. Fairly straightforward, using what you saw under the microscope. Okay. Explain what you think happened to enlarge the specimen. So I'm going to want an explanation here that involves the path of the light as it goes through the microscope. Okay? Don't just say it made it bigger. I know that. That's painfully obvious. Okay? Tell me how it made it bigger. Okay, guys, listen please. Describe and explain the steps you took to find and focus your paramecium. All I'm looking for there is give me a brief explanation of what you did. I put the slide in, then I did this, then I did that. Okay, give me kind of a brief rundown of how you got it to work. And question number 3, we talked about a little bit already. Why do we add up water to a wet mount? Want a pretty good detailed explanation of why that works and the effects it has. Okay, then same as always, state your conclusion, okay, and restate your hypothesis, and then 2 sources of error with ways to fix them. Questions on what you're doing? Okay, so that's going to be due on Tuesday, March 22nd. Right? You can get a pretty decent start on it today. Tom's question. Yes. Yeah. Yeah, just like you did for the planning for the toxin project. Same thing.
Like the planning one.
Right.
I took it on my phone and then I had to show you because I got
Right. We'll have to look. If it doesn't work again, come in and see me before it's due, and we'll make sure. There's just usually one or two little things. Okay. Any other questions on that? Okay, you got some time to get working on it, so do so.